Yeah, hello. Karen, is that on you? Yeah, people are coming. Welcome again. Welcome all as we log in. Just check in and tell me, Madam, we are here. Welcome all. Welcome for the metabolism class. We are doing metabolism, only for metabolism class. Just do the chat and tell me if you are there. One person already there, just tell me who you are. Then I'll be able to say hi. Yeah, welcome. Thank you for the subscriptions, for the likes. Thank you so much. Let's keep on subscribing. Let's keep on coming live. We are having a metabolism class, both nursing and oral health, biochem 2. Both are biochem 2. But we have nursing and oral health group who are doing metabolism. Welcome all. Even everyone who just want to listen, you're welcome. Good. Thank you, Emmanuel. Please say that you are there. Ian, welcome. Thank you. Irene, Mikali, thank you so much. Yes, somebody saying, hi, madam, I'm present. Welcome, welcome. Tell me OH or BSN, all are welcome. The OH are doing it as biochemistry too. I requested them to join, whoever want to listen, because we already teach them in class. So if they want to catch up with the concept, they are free to join. Yes, Cindy, thank you so much. Welcome. Shama, I can see you. Welcome. Football news. Yes. Somebody Q30 present. Welcome. Thank you so much. Yes, Harriet, welcome. Ian, OH. Yes, is it the OH or O? It's still okay. Welcome. Sabari, yes. Very welcome. Tell me the OH. Oh, yes, the OH. Sorry, Ian. Yes, welcome. That is the oral health. I want them to be represented here because I told them they had to join. Oh, sorry. OH, yes, now I can understand. I thought the OH of hydroxyl group, and I'm the one who have said, yes, Karen, welcome so much. Let us keep on logging in, please. Sharon Eboso, yes, hi. Welcome all, welcome. We need to learn together. Just keep on telling your friends. Let's log in so that I don't take much time, just as I also take 1 hour. We just need to go 1 hour, but then bit by bit so that we can understand. Lokale, hi, welcome. Welcome all, welcome. Zakaria, yes, present. Abdullahi, OH, I think is saying OH present. Kabote, it's okay. But welcome, Abdullahi. Roda Kemto, hi, Simiyu. You are most welcome. Please let us keep on logging in. We log in. You can log in through the channel. If you don't have the email address, you can log in with a friend. It's still okay. We'll learn together. I don't think I've left someone out. Keep on logging in. I think we are still at 14. I can see 14 people. I don't know whether it's showing the correct one, but let us keep on logging in. And then, good. Thank you so much. 6 people at a go. They've come. Tell me who you are. Then I can say hi. From my end, I can say hi kindly. We'll be able to start maybe 5 minutes later as we continue, but then we'll be able to revisit whatever we did last time and then the new things which we need to do today. And I hope it will be helpful to the students, especially the nursing students. My student whom I taught one, so I thought of helping out as well for two. I hope it will be able to be of help to the students. You will tell me the feedback after the lectures or even before. Or even in the chats, you can tell me, Madam, the other time I didn't understand this part. As much as I'm going to revisit, then I'll be able to answer. Because when I start giving the lecture, I don't look at the chat, so I might skip a comment. Yes, it stopped at Barak Simiu. I welcome you all. I still give 4 minutes to go because I need just only to take 1 hour. I don't need to take much of your time because you already have a lot to study and the weekend is also ahead, only tomorrow. Then we embark on classes. But I appreciate. We have to make an effort for us to pass. As I also dedicate my time, the students have also dedicated their time. There is no way the students can't pass. So we'll have to pass together. We are going to work together and we shall achieve it. So as we wait for other people to come in, because maybe they're still taking time as they come in, because they have to have the net, the Wi-Fi or the internet. But let us revisit. I don't see anything. I wanted somebody to say, especially the nursing, what the challenges were, if it were okay, whatever we discussed last time. But now, thank you so much. Thank you so much for the likes. Together we can. Huh? It will be fine. Don't get worried. In life, don't get worried. I always tell you, don't make it complicated. Don't complicate it. If you complicate it, then everything will get complicated. So what I want to say is, I want us to revisit last week. And while I'm revisiting whatever we did last time, yes, Andungu, there you are. Hello, I can see you. Welcome. And today also accommodate Cheryl. The last time Cheryl was hanging on you. So yes, good to see me. Thank you so much. Good to see you too, my friends. So I'm saying that I have to revisit whatever I did last time. As we come in, I'll be able to see and welcome everyone, but let that, oh, thank you. We are now at 32. Thank you so much. We have both oral health, who are doing biochem 2, as well as nursing. Betty, oral health, are you there? Just tell me in the chats, because I've seen, oh, Cheryl is with you. Thank you. I've seen Karen. Oh, Ngor. Thank you so much. Yes, Madam Winnie again. Madam Winnie again. Ngor, we are in biochem 2, but you can just listen. Thank you, Senior. You are from oral health. So we have to revisit what we did last time, because with the nursing, Precious Minor, hey, hello. So whatever we did last time, the oral health were not there, but I already did it in class. So we can just do a quick recap of whatever happened last time. We said that at the point glycolysis takes place, or at the point glucose oxidation takes place, it finds when digestion is already done, absorption is already done, and then now it goes to glucose oxidation or breakdown of that glucose. But then we also mentioned that at that point we're talking about the fed state. So there is a lot of glucose. Some have to be broken down, the 10 steps which we saw, the 3 stages which we saw, and then we have to generate energy in form of ATP. At that point, we're talking about D-glucose. We said optical specificity of enzymes. The ones we are working on carbohydrate metabolism have to be the D form of those sugars. So they are going to break our D-glucose to pyruvate, 2 of the pyruvates, with the main objective of generating energy in form of ATP. So we also saw the 3 stages. We said And this one I discussed with the class, the OH. We're just reminding ourselves whatever we did with nursing, because before I can proceed with whatever we are going to do today. So we said stage 1, as you start with your D-glucose, you are going to destabilize it such that at the end of stage 1, you have fructose 1,6-bisphosphate. Stage 2, we said we are generating the 2-trioses, the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. That is the main objective of stage 2. But then the dihydroxyacetone phosphate will go to lipid metabolism, especially the synthesis of cholesterol. But then the glyceraldehyde 3-phosphate will go to stage 3. The main objective of stage 3 was to generate energy in form of ATP. So we mentioned that there is 3 irreversible steps. Step number 1, where the exokinase is irreversible. Step number 3, where the phosphofructokinase does its work, is irreversible. And lastly, step number 10, pyruvate kinase is irreversible. So we mentioned those irreversible steps such that at the time we'll be looking at the body is starving, there is low glucose, then the irreversible steps, we are talking about gluconeogenesis at that point, the irreversible steps will be bypassed. It means we'll have new enzymes there. So we talked about the 3 irreversible steps. But you have to bear in mind that at that point, insulin as a hormone regulates glycolysis. It means as much as some glucose are converted to pyruvate with the main objective of generating energy in form of ATP, some of glucose are converted to glycogen. So we have glycogenesis also going on. So glycogen, we need to have enough glucose, which is stored in form of glycogen. But this glycogen is always not enough at the point of starvation, because it only provides glucose for 24 hours. So the body has to look for glucose from the non-carbohydrate precursors like glycerol, like amino acids, like lactates. So whatever we are mentioning here is at the point that glycolysis is taking place, the insulin regulates that pathway, and the 3 main enzymes which are irreversible are the ones which control the whole pathway of glycolysis. So today we are moving to the fate of that pyruvate. Where do the pyruvate go? So depending on aerobic, on anaerobic, my oral health students, whatever I'm doing here is the notes which I gave, the slides which I gave. So you can check on them as I move, because I don't know how to project on YouTube, but you can put it on a laptop with a friend and then you just follow as I explain those slides. So I don't know how to project. That's why I'm referring to whatever we have. So the notes which I gave you in class, you can open maybe in a tablet, on your phone, but then as much as you can listen, we'll be able to communicate. We can just cope. So I'm saying the fate of pyruvate. After pyruvate has been formed in that main pathway of glycolytic pathway, or the main pathway of glycolysis, it has anaerobic or aerobic, based on aerobic or anaerobic state. Aerobic, you know, the presence of oxygen. And then anaerobic, absence of oxygen. So in the presence of oxygen, our pyruvate is converted to acetylCoA. The acetylCoA, which it is converted to, is very important in lipid metabolism and also an important material in the tricarboxylic acid cycle. The entry point in tricarboxylic acid cycle or Krebs cycle is the acetylCoA. Without the acetylCoA, the Krebs cycle cannot be complete. So we get the acetylCoA from glycolysis, that is from pyruvate in aerobic state, where we have the formation of acetaldehyde. The first step, pyruvate is converted to acetaldehyde, but then there is a removal of 1 carbon. So there is decarboxylation. Most of these enzymes get the name from the reaction. Nursing students, you can remember. Still, the classification of enzymes, specificity of enzymes, where the decarboxylases were, you could remember very well. So decarboxylation from the reactions, that is how those enzymes are classified. So they are removing a carbon at that one step, and you are forming acetaldehyde. From acetaldehyde, it's going to form ethanol, sorry. So that is in anaerobic state. We are talking about the formation of ethanol. So we have the pyruvate, decarboxylase, removing a carbon, presence of or absence of oxygen, sorry, it forms acetaldehyde. Then from acetaldehyde, it forms ethanol. But that one only works in the microorganism because it's fermentation. So we talk about anaerobic state because there is no oxygen. Pyruvate converted to acetaldehyde, removal of one carbon, decarboxylase catalyzing that step because there is removal of carbon. Then from acetaldehyde, it forms ethanol in anaerobic state. So absence of oxygen. So the alcohol dehydrogenase will catalyze that step where the ethanol is forming. So that one always takes place in the yeast or some of the microorganisms because it is a fermentation process. We still talk about the fate of pyruvate, but in this case, we are talking about anaerobic because it's going to form ethanol. Another state where it forms lactate, it's also anaerobic. In absence of oxygen. In absence of oxygen, it forms lactate. And lactate is a source of energy for some of the brain cells as well as the cardiac muscles. So at that point, we are talking much about in anaerobic state, our pyruvate from the glycolytic pathway in the absence of oxygen can be converted to lactate. Just one step. Lactate dehydrogenase, catalyzing that step. And in anaerobic state, both enzymes, the alcohol dehydrogenase, the lactate dehydrogenase, those are oxidoreductases. If you could remember the class of enzymes, they catalyze oxidation reduction reactions. So most of them will be the dehydrogenases, and they have the NADH, the NADPH, and the FADH2s. So you'll be able to relate them as oxidoreductases. So we are talking about dehydrogenases in the anaerobic state where the oxygen is absent. Alcohol dehydrogenase, where you could see pyruvate forming the acetaldehyde to ethanol, that alcohol dehydrogenase is NADH dependent. Whatever goes in is what the enzyme depends on. So even if you look at the lactate dehydrogenase is NADH dependent, then it releases NAD+. But when you look at the pyruvate to be converted to acetyl-CoA, where the enzyme pyruvate dehydrogenase complex of enzymes, in the presence of oxygen, it is going to form acetyl-CoA. So I say this enzyme, pyruvate dehydrogenase complex of enzymes, it involves 3 enzymes there. But you will see, instead of it being NADH dependent, it is NAD plus dependent, the opposite of the ones without the oxygen. So it is going to form the acetyl-CoA. And the acetyl-CoA is the entry point to the TCA cycle. And that's why we are talking about the amphibolic role of glycolysis. It is going to provide materials for the other pathways. I know even the acetyl-CoA can come from the breakdown of the saturated fatty acids, especially the even-chained fatty acids. But then basically, how it is going to connect to the TCA cycle is through the pyruvate. So the pyruvate in aerobic state, presence of oxygen, will be converted to acetyl-CoA, the enzyme pyruvate dehydrogenase complex of enzymes or pyruvate dehydrogenase enzyme. Then it is going to convert it to acetyl-CoA. The acetyl-CoA will enter the TCA cycle. We'll be able to talk about the TCA cycle next week because we are going bit by bit. It could be even the other week because I'm planning to do fructose, entry of fructose to glycolysis and galactose, then I stop. So that the other week we can look at gluconeogenesis. So if time allows, we can introduce TCA. But for now, what we have to know is the fate of pyruvate. Where do the pyruvate go? Based on the aerobic state, presence of oxygen, anaerobic state, absence of oxygen. In anaerobic state, it forms lactate as well as ethanol. Ethanol fermentation for some of the microorganisms and yeast. The process will take place there. Then for the lactate, which is used as a source of fuel for the brain cells and the muscles, the cardiac muscles, then that's how important those ones are. But then it has to come from the glycolytic pathway. So that's how important it is. It's going to provide materials for the other pathways. I still mentioned the 3-phosphoglycerate, which you could see was now converting to 2-phosphoglycerate at the point we had the phosphoryl shift where the mutation was taking place. That 3-phosphoglycerate is the material for synthesis of serine. You know, serine is alcohol-containing amino acid. The dihydroxyacetone phosphate, still an intermediate of the glycolytic pathway. I'm already at the medical and biological importance of glycolysis. Some, it's easy to read on your own. That's why I'm stressing on the intermediates. The dihydroxyacetone phosphate will go to lipid metabolism. Some, like 1,3-bisphosphoglycerate, is converted to 2,3-bisphosphoglycerate, which is used much in oxygenation of the erythrocytes. Glucose 6-phosphate, which forms in the first step where the exokinase is used as a material as well in the pentose phosphate pathway, the best pathway where the nucleic acids, the ribose, which form part of the nucleic acids, are synthesized. So the glucose 6-phosphate from the glycolytic pathway, even from glycogen breakdown, will be used in the pentose phosphate pathway. So the amphibolic role of glycolysis, it is a core in these pathways. It provides material for all those. And I told you, you have to know the glycolytic pathway for you to understand the other pathways. It will be very easy if you understand the glycolytic pathway. Already we did structures in Biochem 1. Now we are trying to get the correct enzymes. And I told you that it is better you leave out not writing the enzyme. By writing a wrong enzyme, a reaction where the kinase does its work, and then now you are writing an oxidoreductase. That is totally wrong. As long as you write the intermediates are all correct, and then you have a wrong enzyme, that is totally wrong. So whatever I'm trying to stress much is the amphibolic role of the glycolytic pathway. Another main role here is for fructose and galactose. They use the same pathway, glycolysis, to provide energy. So that is what we are going to look at. Because as much as you see fructose, which comes from sucrose itself, and remember sucrose was a disaccharide, and I said in the small intestine, the sucrase will break it down to the simpler monosaccharides. And I will always tell you specifically, tell me the alpha D glucose and beta D fructose being the components of sucrose. So the glucose, it will be broken down simpler into D glucose. The glucose itself will go to glycolytic pathway and go through the 10 steps and provide energy. That is fine. But what of fructose? Fructose also has to provide energy. So we have to generate an intermediate of the glycolytic pathway, is when we can say to glycolysis. As much as you can see any of the intermediates of the glycolytic pathway, when you start with fructose, D-fructose for that matter, then you will just show an arrow, it goes to glycolytic pathway. So for example, after those absorption, and I said absorption of fructose is very slow because of the keto group at carbon atom number 2. It gets absorbed by facilitated diffusion. The other ones are by active transport, very fast. So facilitated diffusion, very slow than the aldoses. But at last, it has to be absorbed. Then after absorption, fructose has to provide energy, just in form of ATP as glucose. So our fructose, remember, we did substrate specificity of enzymes. And that is what you couldn't see for the glucokinase, because the hexokinase has high affinity for glucose than the glucokinase. So in the case of fructose and galactose, the substrate specificity will be seen. So fructose, we have the fructokinase. Still a phosphoryl transfer step. So the fructokinase will be specific. For D-fructose, still we are talking about optical specificity of enzymes. The ones which are involved in carbohydrate metabolism, they are working on the D form of the sugars. So we have D-fructose. Then we have fructokinase, substrate specificity. It is specific for fructose. It is going to transfer the phosphate to the first carbon atom of fructose. And you'll have fructose 1-phosphate. The kinase still, it will go in as ATP utilization step and go out as a diphosphate because 1-phosphate will be used to phosphorylate our fructose at carbon atom number 1 to form fructose 1-phosphate. So at that point, you're talking about the fructokinase. Remember, I said for the kinases, we need magnesium ion or manganese for it to be active at the active site. So it has to be there. So after forming fructose 1-phosphate, you know, it has several options. Fructose 1-phosphate, the first route is fructose 1-phosphate can be converted to fructose 6-phosphate. That is phosphoryl shift. You shift the fructose from, shift the phosphate from the first carbon to the sixth carbon. Then fructose 6-phosphate is an intermediate of the glycolytic pathway. Then you give an arrow, it can go to glycolysis because from fructose 6-phosphate, it's going to form fructose 1,6-bisphosphate. Then you'll have the aldol cleavage, then down the line to stage 3. So you will finish the end of stage 1, you'll enter stage 2 to stage 3. Because we are looking at the ATP generation steps. And I mentioned that step 7 and step 10 of glycolysis are the ATP generation steps. You talk about substrate level phosphorylation steps. So it means the first option is our fructose has to be converted to fructose 1-phosphate, fructokinase catalyzing the step, ATP coming in, triphosphate surrounding the adenosine, out as a diphosphate because 1-phosphate has been used to phosphorylate our fructose at carbon atom number 1. So the first option is phosphoryl shift, mutase. We'll shift the phosphate from the first carbon to the sixth carbon. Then you just give an arrow to glycolysis. The second option is our fructose will be converted to fructose 1-phosphate. Then aldolase, the aldol cleavage, it cuts it into half. And when it's cut into half, the upper part will be dihydroxyacetone phosphate. And then the lower part is glyceraldehyde. Not glyceraldehyde 3-phosphate so that you can stop and say that it's an intermediate. No. I repeat. Option number 1, how fructose can provide energy or how fructose can enter glycolysis. Option number 1 is it's converted to fructose 1-phosphate via the kinase. Phosphoryl shift. Let mutase do the work. I told you, even if you tell me mutase without mentioning fructose 1-phosphate mutase, I'll give you a mark. Just as the same as I talk about phosphoglycerate mutase. If the name phosphoglycerate mutase is longer and you can't remember it so that you can write your own, if you just give me mutase, I'm entitled to give you a mark because you know the type of enzymes, the correct enzyme, class of enzyme which is catalyzing that reaction. So in this case, I said the first option is for fructose to provide energy in form of ATP, we need to get an intermediate of the glycolytic pathway. So the intermediate of the glycolytic pathway for the first option is after converting our fructose to fructose 1-phosphate, where there is phosphoryl transfer, then the second step will involve phosphoryl shift, a mutase. We'll shift it from the first carbon to the sixth carbon, then give an arrow, say, to glycolysis. The second option, After formation of fructose 1-phosphate, let aldol cleavage take place. It is going to cut our fructose 1-phosphate into half. The upper part will be dihydroxyacetone phosphate. The lower part will be glyceraldehyde. You can't stop there. The enzyme is still aldolase because it's aldol cleavage. From glyceraldehyde, it has to be phosphorylated. So you have to talk about triose kinase because it is a triose. Kinase are the ones who are doing the phosphorylation. So the ATP has to come in, goes out as diphosphate, so that the phosphate is used to phosphorylate our glyceraldehyde at carbon atom number 3. And you have glyceraldehyde 3-phosphate. Then you show the arrow to glycolysis. Because this question always do not have a lot of marks. If it goes too much, I can give it 5 marks. Using structures, correct enzymes, it will earn you marks. But now you have several options. You can do it the way where you're forming fructose 6-phosphate and say it goes to glycolysis from fructose 1-phosphate where the mutase does the work, or aldol cleavage. It is cut into half. And then the lower part, the glyceraldehyde, have to be phosphorylated by the triose kinase. And then it forms glyceraldehyde 3-phosphate. And it goes to glycolysis. That's how simple it is. Entry of fructose to glycolysis. It means you are being asked, where does the fructose come from? The table sugar. Everybody in the morning will consume the sugar because you can't have tea without sugar. Already you had a source of starch. You had bread. You had what again? Potato. You had cassava, whichever. And then I said digestion has to start. Absorption has to start. But then those are homopolysaccharides. Starch has the amylose and amylopectin. But the most important thing here is in starch, we are talking about the alpha D glucose. So it has to be broken down simpler to the D glucose. For some people, they will show the Howard projection. So it talks about it has to be an alpha D glucose. It can't be a beta. Beta is cellulose. So it can be the cyclic form where the glycolysis starts, but then it is D glucose. So if it's Howard projection, it has to be alpha, and you know how alpha is. The OH has to be below at carbon atom number 1. But the most important thing here is you already had the component of the carbohydrates in the morning. That's starch. Already digestion, absorption, now glucose oxidation. It provides energy. But the sucrose, which you also added in your tea, has to be broken down simpler into alpha D glucose and beta D fructose. So the D fructose we are talking much about in just linear form, the D fructose, that is what we are talking about. So it also has to provide energy by having the fructose 6-phosphate forming, which is an intermediate of the glycolytic pathway seen in stage 1. It will proceed to stage 3. Or it can also form glyceraldehyde 3-phosphate through the formation of, from glyceraldehyde, triose kinase, converted to glyceraldehyde 3-phosphate, seen in stage 2, it proceeds to stage 3. Because we are looking at how do fructose provide energy. The main objective of stage 3 is to provide energy. How do they reach stage 3? Same with lactose. The milk component, the milk sugar. You already taken milk. Most of these things you can't miss a meal without a carbohydrate because they are the energy giving foods. The lactose has to be broken down. Still in the small intestine, lactase is there. Only for the people who have lactose intolerance. They can't consume something which has milk in it. But the lactase will break it down to beta D galactose and beta D glucose. But as much as we're talking about the beta D glucose, beta D galactose, where do our galactose go? Simple. It has to have an intermediate of the glycolytic pathway, then you stop. So that is how fructose provide energy. For galactose, it will start in Howard projection. It will start with the galactose itself. Then the galactose, the galactokinase. We are talking about specificity of enzymes. Substrate specificity of enzymes. It will be specific. The first enzyme, which will phosphorylate our galactose, it is going to form galactose 1-phosphate. The galactokinase will be specific for galactose. And the three enzymes are very important for galactose entry to glycolytic pathway. That is galactokinase, which phosphorylates, uridyl transferase, which activates our galactose, and then lastly, epimerase. Because remember, galactose and glucose are epimers at carbon atom number 4. So the galactose at some point has to be converted to glucose. Because in the glycolytic pathway, we don't see any trace of galactose, but we only have glucose. So we have to convert it to something similar in the glycolytic pathway. And now, the first step, after we've had our lactose, the milk sugar, we need the galactokinase, specific for galactose. We are talking about phosphoryl transfer. Still, the kinases need magnesium ion or manganese on their active site. Galactose will be converted to galactose 1-phosphate via the enzyme galactokinase. Very important. It is the one for phosphorylation. Then the activation is addition of uracil or uridine. The presence of uracil there, very important. Even in glycogen synthesis, activation is very important. Uracil will still come in. So at this point, there is activation of galactose, and it has to be activated for it to provide energy. So the first step is galactose has to be phosphorylated. By adding a phosphate, you form galactose 1-phosphate. The next step, where you have the uridyl transferase, you have to activate our galactose for it to provide energy in form of ATP. So the enzyme is galactose 1-phosphate because the substrate is galactose 1-phosphate, uridyl transferase. So there is a glucose which comes as a carrier. It carries the UDP, the uridyl diphosphate or the uridine diphosphate. At that point, it acts as a carrier for the activation. So it carries that component which is going to activate our galactose 1-phosphate. And then after activating, it forms UDP, galactose. Then the glucose will go with 1-phosphate, which was on our galactose 1-phosphate. So the glucose 1-phosphate which goes out, because the glucose can't go alone. As much as it came to ferry the uracil so that our galactose can get activated, the phosphate which was attached at carbon atom number 1 of galactose will have to go with this glucose which came in as a carrier. So it goes out as glucose 1-phosphate, but nothing gets wasted. The glucose 1-phosphate can be converted to glucose 6-phosphate. That is phosphoryl shift by mutase, so that glucose 6-phosphate can provide energy in form of ATP in glycolytic pathway. But let us not lose focus. Let us go to the main pathway. After formation of galactose 1-phosphate, it is going to be activated and you will form UDP galactose. The enzyme there is uridyl transferase. It is the one which is responsible for activation. The other core enzyme is epimerase. Those three enzymes are core. If one of them is defective, it leads to galactosemia. And galactosemia is very serious. You know, the liver will be affected because it plays a role. By the time you're looking at the liver function tests, the function related to carbohydrate metabolism, you look at how long it takes for the liver to release galactose from the system. If it delays, then it means there is a problem with the liver. And at that point, you're talking about one of the three core enzymes there is a problem. It could be the kinase, it could be uridyl transferase, or it could be epimerase. So the third one, which is very important, is UDP galactose epimerase. It just talks about 4 because they are epimers at carbon atom number 4. And I always say, even if you don't mention 4, I don't think whether it can be wrong. As long as you know it's epimerase, it's going to convert our galactose, UDP galactose, to UDP glucose because they are epimers. So our UDP galactose, the galactose which has been activated, has to be converted to UDP glucose. Already, you've started seeing traces of glucose somewhere. We are leading to a direction where we are going to enter the glycolytic pathway. So at that point, you are talking about epimerase converting our UDP galactose to UDP glucose. Then a phosphorylase will remove the uracil now after it has done its work. So it will come in with pyrophosphates, 2-phosphate, but then will release UTP, uridyl triphosphate, and then we'll have glucose 1-phosphate. It is easy to detect the next step because it will be phosphoryl shift. Our glucose 1-phosphate will be converted to glucose 6-phosphate via the mutase. Then the glucose 6-phosphate, you'll say the arrow goes to entry of glucose to the glycolytic pathway. Because from glucose 6-phosphate, it will form fructose 6-phosphate. It will proceed to the end of stage 1 where we form fructose 1,6-bisphosphate. It will proceed to stage 2 to stage 3. But there is no need of explaining all those steps. After identifying an intermediate of the glycolytic pathway, then you stop. You just show the arrow to glycolysis, and that is how it works. So the entry of fructose, the entry of galactose to glycolytic pathway, very important. At your free time, read the medical and biological importance of the glycolytic pathway, which I skipped up here. Very important. As you look at each of those intermediates, you'll realize how important the glycolytic pathway is. It talks about production of energy in form of ATP based on anaerobic state. Anaerobic, we say it's going to form ethanol and lactate. Under aerobic state, it's going to form acetylCoA. So at that point, it will provide energy because the main objective is to provide energy in form of ATP. So you'll see how important it is. And then there is also a point which talks much about the formation of 2,3-bisphosphoglycerate for the oxygenation of the tissues. So at that point, you are talking about the formation of 1,3-bisphosphoglycerate because you don't have 2,3 there. 1,3-bisphosphoglycerate is an intermediate of the glycolytic pathway. It is going to form 2,3-bisphosphoglycerate because only there will be a carboxyl, C double bond O and O minus, shifting from carbon atom number 1 to carbon atom number 2. So you talk about 2,3-bisphosphoglycerate. Mutase is going to do the work. So instead of you having 2 of the phosphates at carbon atom number 1 and number 3, now you'll have 2,3. So that component is very important for the oxygenation of tissues. That's very important. So we talk much about how important glycolytic pathway is. It talks about fatigue for the PFK. If the enzyme is having a problem, then there is fatigue for the muscles. So the PFK regulate the whole pathway. It controls the whole pathway. Another point says dietary fructose and galactose use the same pathway to provide energy, just what we've discussed. And then it also mentioned the amphibolic role of glycolysis. For example, it talked about the formation of lipids and amino acid. If you talk about the formation of amino acids, pyruvate, we are going to talk about glucogenic amino acids. Pyruvate can be converted to alanine, one of those amino acids which you know fall under the aliphatic. So it's also a way where it can be used in formation of some amino acids, formation of lipids. via transamination. So if formation of lipids, you have the dihydroxyacetone as well as the glyceraldehyde 3-phosphate. So we have the 3-phosphoglycerate, which also helps in the synthesis of serine. It just shows how important the glycolytic pathway is. And then another point it says, it gives us the acetylCoA, which has to be an intermediate for the TCA cycle or the Krebs cycle. Very important. At the entry of the TCA cycle, the main objective of the TCA cycle, you're going to see the generation of the reducing equivalences. By reducing equivalences, I talk about FADH2s and NADHs. They are going to be used as electron carriers in electron transport chain. You talk about the oxidative phosphorylation. So the TCA cycle is going to provide the reducing equivalences, NADH and FADH2s. It can't provide that one without the entry of acetyl-CoA, which comes from the pyruvate in aerobic state. And still, the TCA cycle, we talk about some of the amino acid also uses at the entry point. So the TCA cycle will also connect the amino acid metabolism or protein metabolism with the carbohydrate metabolism. So the entry point of the TCA cycle is the acetyl-CoA, which is provided by the glycolytic pathway through the formation of pyruvate. It has to be converted to acetyl-CoA in the presence of oxygen. So that is how important this pathway called glycolysis is. And even at the point it talks about the glyceraldehyde 3-phosphate is also used in synthesis of cholesterol, also used in the synthesis of some of those vitamins like thiamine. So at your free time, you'll go through it and you'll be able to understand. I want us to absorb things slowly by slowly. I said I'm not going to move so fast. Karen, I don't know how far you've got. Samrat Basu, how does the amino acids form? Samrat, your question says, somebody is asking here is where? Oh, Nakaya, I'm here, though it's raining heavily. Welcome. Now, I need to answer Samrat Basu. Samrat, we are going to look at gluconeogenic amino acids. At the point when you talk about how the amino acids form, you're talking about the synthesis of the amino acids. So, in the case of the glucogenic amino acids, you are talking about from pyruvate, they can form the amino acids. We are going to talk about the gluconeogenic pathway. If you talk about gluconeogenesis, you are going to see how those amino acids help match in synthesis of glucose. That is different from whatever you are asking. At the point when we reach synthesis and degradation of amino acids, you'll understand well that the reverse and the forward reactions help us to understand. Somebody like aspartate, aspartate, Samrat, aspartate. Aspartate falls under the acidic amino acids. And aspartate can be formed from oxaloacetate. And oxaloacetate is an intermediate of the TCA cycle and as well as in gluconeogenesis in between here. So the oxaloacetate will be forming aspartate. Aspartate can also be broken down to form oxaloacetate. Then asparagine can be broken down to form aspartate by deamination, and then you have the transamination taking place in between here. You form oxaloacetate. The other way around. Glutamate is going to form alpha Kg. So several amino acids. At the time we reach degradation and synthesis of amino acids, Samrat, will be able to clarify how those amino acids are forming. Next week, Samrat, I'm doing gluconeogenesis. Gluconeogenesis is not just reverse of glycolysis, but we are going to look at how the arrow goes, because now the body has low glucose. We are talking about its own starvation. And that is why you can see people who are in areas where there is famine. Famine is the one which deals with food. they don't die that fast because the glycerols will keep them going, the lipids. And even at that point, when you talk about starvation, the glucose level is low, the amino acids like alanine is going to be converted to pyruvate. We are going to look at gluconeogenesis next week. Very interesting. The conversion of alanine to pyruvate, the arrow goes towards pyruvate from alanine. We are breaking down that amino acid so that it can provide us with glucose. Then it goes, there is an intermediate in between here, oxaloacetate. Then we have the phosphoenol pyruvate. Because at this point, we're talking about the bypassed steps. You know, we are talking about the 3 main steps, which are irreversible. They are the only ones which are going to be bypassed. So from step 9 to step 6, yes, it is still going to be the same enzymes. The reversible steps are going to be the same. Enolase is still enolase. Mutase is still mutase. Glycerate kinase is still glycerate kinase. Glyceraldehyde 3-phosphate is still glyceraldehyde 3-phosphate dehydrogenase. But then the arrow goes towards formation of glucose. Now we've used amino acid as an example because the body has to look for ways on how to provide energy in form of glucose from the non-carbohydrate precursors. And if you talk about the non-carbohydrate, amino acids are not carbohydrates. So that the non-carbohydrate precursors, glycerol, the formation of glycerol, you see where it connects the dihydroxyacetone glycerol, it is going to connect at that stage 2 of glycolysis. Then the arrow goes towards formation of glucose. So the formation, the synthesis of amino acid, we are going to find several of them. But what I mentioned here was 3-phosphoglycerate, which is found in step 8, where the mutase is. It forms 1,3. The 3-phosphoglycerate, which you are seeing there, is the one which is used in synthesis of serine. That is what I mentioned. So an intermediate, the glycolysis, how important it is to protein metabolism. It can be used. It can provide materials for synthesis of some amino acids like serine. Yes, Senior is asking. I'm trying to thank you, Samrat. I'm happy that you are satisfied. Madam, in an exam, when we are asked to use structures, do we use the linear structure or the cyclic form, or we can opt for any of them? A very good question. When I looked at the nursing, official GIT, GWhite, I don't know how to call you. Are you in oral health or nursing? Because I need to specify something. The ones in nursing, from the PDF file, which I saw, the lecturer started with the cyclic form. But as we proceed, we have the linear form. The way that it is presented, in my case, in carbohydrate, you can see, in glycolysis, I use the linear form. Because the overall note here is we have to break it to a simpler form of D-glucose. So if it appears in linear, it means it is D-glucose. If it appears in cyclic, it means we have to note about the position, whether it's alpha or beta, but look at the position of the OH, how the lecturer presents the notes. Whatever you saw in galactose, everything was in cyclic form. But it's still the simpler form. It is the simpler form of those sugars. That is the most important thing. The simpler form of those sugars, be it cyclic, be it linear. So for the galactose, the notes which you can see, as far as I'm concerned, it is a cyclic form. Use the cyclic form which you're seeing there. For the nursing students, what the lecturer gave was it starts with the cyclic form. And even for some sites, it starts with the cyclic form. But the most important point here is, it is no longer the polymer. It is no longer the disaccharide. You have the simple form. We talked about the simple sugars. You can't break them further, because that is now what is broken down to provide energy. Now it can play its role of providing energy. You talk about glucose generating energy. How? So it is in simpler form. So official light, I think it's light. If you are in oral health, I gave mine in linear form, especially glycolysis. Give me in linear form. And in linear form is where I can easily see. I told you, drawing those structures, don't cram anything, master. Because it's just a change on one carbon. If you draw it in linear form, you can clearly see the change. At carbon atom number 1, we had the aldehyde group. You know very well our glucose from Biochem 1, the aldohexoses. At carbon atom number 1, we have our D-glucose. You know our D-glucose, the way it is an hexose, naming of monosaccharides, 6 carbons down the line. Functional group, at carbon atom number 1, we have our aldehyde group. At carbon atom number 6, it remains CH2OH. But since it is a D-glucose, we talked about optical specificity. The OH at carbon atom number 3, it lies to the left. Balance. 2, 4, and 5 to the right. You know it is D-glucose we are dealing with. It is the simplest form of those sugars. You will clearly see they change at carbon atom number 1. A phosphate is added. The kinase does the work. Of course, the hexokinase has high affinity for glucose than the glucokinase. So the hexokinase work there. You will just see from carbon atom number 2 to number 6, everything is the same as whatever you do with the first one. Only add the PO3 minus 2, the phosphate. CH2, add the phosphate group. That's how simple it is. Then you go to the next one. You know the functional isomers. It finds when you already know the structures. In biochemistry 1, you know the structure of fructose. D-fructose to be specific. A ketohexose. Keto group, carbon atom number 2. Number 1 has CH2OH. The last carbon, CH2OH. Now, since you know it is fructose 6-phosphate, just put a phosphate at carbon atom number 6. Carbon atom number 1 and number 5 remain the same. Then move to the next step. Fructose 1,6-bisphosphate. Nothing changes from fructose structure which you had, fructose 6-phosphate. Only at carbon atom number 1, just add a phosphate. So if you draw it in a linear form, for me, I'm saying for myself, in my glycolysis, use the linear form. We are talking about the Fisher's projection. It will be easier for me. I can easily see the changes in each and every step and how clear the enzymes are catalyzing the steps. If it is in galactose, use the cyclic form, the way it is showing there in the slides. It doesn't cost you a thing. It's only the simpler forms of those sugars. Some lecturers try to present them in cyclic form. If somebody presents the notes in cyclic form, draw it the way it has been presented. Don't come your way. It is fine. It is still the simple sugar. But now some are in cyclic, the other ones are in linear. So, James Locale, let me see. Oh, Locale is saying something. I'm from BSN, correct. Official, you are from BSN. So, just be keen. Oh, somebody's asking, have same structure breakdown. Let me just finish with official. Yes, official, it's good you've mentioned you're from nursing. So make sure what Dr. Nyaga gave in the notes, what I saw was starting with the cyclic form. Start with the cyclic form. Just look at how the changes are. For each of the carbon, what you'll notice only a change in one carbon. Nothing much. But make sure you get the correct names of the enzymes for those reactions. Very important. Somebody saying something. In stage 2, step 4 and 5, have same structure breakdown of glucose. Senior. Stage 2. Stage 2, we don't have glucose. Am I not understanding? Senior, stage 2, no glucose. Stage 2, to be precise, the main objective of stage 2 is to generate the 2-trioses. Senior, let us be clear in this. So, in stage 2, the 2 processes which takes place there is only in step 4, you have the aldol cleavage. We are cutting. we are cutting our fructose 1,6-bisphosphate into half, 50-50. The upper part, dihydroxyacetone phosphate, the lower part, glyceraldehyde 3-phosphate. Then, step 5, functional isomers, whatever we discussed in biochemistry 1, of the same molecular formula. They are both trioses, C3H6O6. But the difference in structure is based on the difference in the functional group. So we are interconverting a ketotriose to an aldotriose, an aldotriose to a ketotriose. So there is an isomerization step there. Then the names come, triose phosphate isomerase. So in stage 2, we don't have glucose. Good. Let me answer Locale. Locale is saying, to understand the way you break down difficult concepts and connected them to real-world application truly enhanced my learning. Thank you, Locale. We shall pass. What I tell you to do is read and understand. Don't cram. And try to imagine, when does glycolysis takes place? When do beta oxidation takes place? When do amino acid breakdown take place? When do gluconeogenesis takes place? You will find it very simple. And imagine one of those enzymes getting defective. The condition. There is already a problem. And that is what we do in Biochem 3. Clinical biochemistry. These enzymes you see in these pathways, they are proteins in nature. There is a gene which codes for that enzyme, which gives that phenotype, that clinical condition. Maybe you can see it, or you have to confirm a test. The enzymes are doing very important thing. And you have to remember that the organelle where all this is taking place is also affected at that time. And you also have to remember, we start from the biomolecules. It can't be complete without the reactions. And it can't be complete without the biomolecules, without the carbohydrates, without the lipids, without the proteins. And remember the buffers and water, very important. Buffer doing the regulation of the physical pH in the system. Such that by the time you go to the hospital that there is a problem, you say, today I'm not myself. Already, there is a problem, there is some turbulence, there is an interference, the acidity or alkalinity of the system, the buffering system. And at that point, you're talking about a metabolic pathway. There is an enzyme which is defective. And then at that point, the enzyme is defective. Then there is something which is of high level, because there is normal level, which makes the normal system to be normal. And then you say, today I'm fine, I can go and do my work, I can go and do my job. You see, those enzymes have to work well. But at that point, they are defective. There is a problem. So all goes to biochemistry, chemistry of life. And it can't be complete without these reactions. So that is why in the introduction I said we have to understand the physiological processes in this system. It could be human, it could be any. If it could be plants, it's another system. But we have to understand, even plants work like animals. They will just be happy when they had the basic. They are watered. They get enough nutrients which they need. They get enough minerals. So it all goes to the processes which takes place. Very important. And the organelle which hosts them. And it has to start from the biomolecules. So locally, I appreciate, but then we have to understand why the process is taking place. Now, why is glycolysis? There is a lot of glucose that the body has to find where to put it. First, it has to have an option of providing energy. Glycolysis will take place. Or the excess ones, glycogen can store it, a granary full of glucose, such that by the time of starvation, when now mahindi imeenda juu, we are saying glucose now is like the mahindi yenye inaiko kwa granary. It's too much. The granary is for the glycogen. By the time on starvation, now mahindi bei yake imeenda juu. Hadi unapata kilo mbili ya unga is 200 shillings. Aya, wenye walikuwa kwa the granary is too small. The silos, I don't know. They'll bring them. They'll be able to provide us with the energy. So at that point, glycogen has to be broken down. Itupatie ile glucose ilikuwa imeweka. So the same enzyme, the enzymes which control the glycogen breakdown are the same enzyme, the same hormone which regulates, the hormone which regulates glycogen breakdown, which we are talking about, glucagon, is the same hormone which regulates gluconeogenesis. Because inatafuta kila mahali, where can I get the glucose? But the glycogen store are not enough for glucose, because even the brain is the one which needs glucose than any other organ. The brain needs energy more. So it needs glucose. So it has to, and you know the work of the brain. I'm not good in physiology, neither am I good in anatomy. But if you imagine the work the brain is doing, until you become sober, you can think, you can remember, you can balance while you walk. All those, the brain needs the huge part of glucose. So when the glucose level is low, we shall talk about that process next week. Gluconeogenesis. Glycogen stores, as much as it stored enough of glucose, it's not enough. It's just going to provide for 24 hours and then it goes its way. So it has to provide glucose from the amino acids. The pyruvate has to be converted. The alanine has to be converted to pyruvate, to oxaloacetate. Aspartate has to be converted to oxaloacetate to provide energy in form of glucose. Glycerols have to come in. So just try to imagine at what state do a pathway take place and how important it is. And if any of those enzymes are defective. Yes. So I've tried the bit I can. For my nursing student, I thank you. For my oral health students, I thank you as well. So you will get back to me. If I get feedback from you, I'll be able to proceed. If you are not satisfied, I'll stop. Because the oral health, I can still teach them in class, but for the nursing, depending on the feedback of the students, we'll be able to proceed. And I hope we'll be able to get something. I told you the lecturers can be different, but we have to cope with all the situations. But what I know, with the students whom I taught in one, we are capable of passing, all of us. And now you can still see the marks. I always tell the students, I can attest, the channel has made tremendous improvement in students' performance. And when I was marking last time, I was very happy because you could see the feedback, how the students will always answer. And I can't say that it's my effort. It is both our effort. Somebody has to put an effort, and the lecturer also put an effort. But I said it starts with total discipline and 100% attendance in class. I will still say that. We have to attend class regularly. And it goes with respect. Just do what brought you to school. And you'll find the university is the best place to be. You'll excel in everything. And then above all, always pray. Just put an effort and always pray. Don't just sit there, you don't do anything, and then you wait for miracles. Miracles will not happen if you don't put an effort. So I know we shall achieve something. Remember, we still have the revision questions at the end of the semester. And for the nursing students, I would love you to tell me by the time you're doing the card how the questions are, so that I can try to speculate how the lecturer sets and possible questions, how we can answer them. That is after the paper has been set, then maybe Karen can forward it to me. If it was written on the board, maybe there is a way you can forward it to me. I see how it was set, so that we can know what you need to answer and where. And maybe it will find when the lecturer has returned the papers and you see what the lecturer wanted. And together, it shall be well. For my oral health, we are still together, so we'll be able to achieve something. but everyone has to put an effort. So I thank you so much, and God bless.